So that was basically chapter 4. In chapter 5, I've gone into the molecular mechanisms of T cell help. Now this is an area where a lot of the so-called cellular immunologists had been focused for at least 10, 20 years. And so there were a lot of people involved, a lot of investigators. And this is a time in the history or in the cellular immunology end of things where everybody who was anybody were doing assays looking at the generation of antibody forming cells. And the question was, what's going on in these things? It was, as I said, you needed to have B cells. B cells then became the antibody forming cells, but they need help. They needed help from the T cells, and they needed help from the macrophages. And it wasn't clear exactly what was going on there. Then in 1971 was the beginning of the dissection in the 70s of what was happening at a more molecular level. And there was a fellow in England by the name of Avrion Mitchison who did a series of experiments and said, it turns out that the only way to interpret my experiments is that the T cells need to have contact with the B cells. And if you separate them by any means and don't allow contact to come, which he called linked recognition, or cognate recognition, which means connecting, then the whole T cell help phenomenon was markedly diminished. But there were also these soluble factors, and people had shown if you could take in or you put in or take out soluble factors, then you would also markedly diminish the generation of these antibody forming cells. And so a bunch of people got involved, and they developed a bunch of different assays for the generation of antibody forming cells, trying to sort out what was going on in all these supernatants and conditioned medium and cognate recognition and so forth. And they had one assay where it looked like they were measuring the proliferation of the B cells, the antibody forming cell precursor cells. And so they labeled this factor B cell growth factor, which I always liked that name because it was a takeoff from T cell growth factor. And then other people were more interested in the antibodies that the cells made, and they could quantify the amount of antibodies coming out and so forth. And so this became then a B cell differentiation factor. And so then there got to be BCDF1 and BCDF2 and 2A and all kinds of things. There's a whole bunch of these things, depending on who the investigators were, and they were all competing with one another. Now, it took a long time to figure this all out, and it took a long time because of several difficulties. The one was that for these assays, the target cells in the assays were heterogeneous. The target cells, for the most part, were mouse spleen cells that had all kinds of B cells in there, but had a whole bunch of different kinds of B cells in there. And B cells are just like T cells in that each B cell would have the ability to bind to and react against only one antigen. So when you took the whole spleen out, you had bajillions of these things. And so the target cells were heterogeneous for the most part. The second problem were the same problem that we had in the whole T cell growth factor IL-2 experiments, and that was that the assays for the proteins, when they tried to fractionate the conditioned medium and separate the different molecules in there, they were cumbersome. The protein chemistry had come of age by then, but it was still an art form, and you had to be really good at the way you did these experiments in order to get any reasonable data. The other thing was is that the assays were not as quantitative as you'd like them to be in order to do the work that needed to be done to separate all the molecules and so forth. And so the heterogeneity of the target cells was one of the problems, but the other problem was that in order to get T cells that would be, if you're interested in T cell help, which is what we're interested in here, the T cells were heterogeneous. They were also coming from a whole spleen or whatever else. And it wasn't until then 1980 that a friend of mine by the name of Max Schreier from Basel, Switzerland, developed the first helper T cell clones. And he developed a whole bunch of, they were all in the mouse, a whole bunch of different helper T cell clones. And so he solved the problem of the T cell heterogeneity by using clone cells. And then several groups worked on the problem of the target cell heterogeneity. And one way around that was to use, nobody had B cell clones or B cell, like we had T cell killer T cells, and then Max had helper T cell clones, but nobody had B cell clones. They tried to make them, but they couldn't get the cells to grow long term. And so one of the ways around that was to use malignant B cells, lymphomas, and they could clone those cells because they grew autonomously and so forth. And on the basis of that, people had come a long way by 1985. Between 1980 and 1985, the fact that they had at least two different kinds. They had the B cell growth factor and they had the B cell differentiation factor. But it wasn't until 85, 86 when a woman from Karolinska Institute in Sweden, Eva Severinsen, she basically duplicated our T cell growth factor assay, except she did it for B cells. But she made a quantitative assay so that you could dilute out all these factors, all these supernatants and multiple doubling dilutions, and so you could quantify what was going on there. And then it turns out that was critical, because then she could test all these different semi-purified factors and so forth from all of her competitors. She was a very nice lady, and everybody decided to collaborate with her. And she then worked out that whatever so-and-so was calling a B cell growth factor and another person was calling it B cell differentiation factor were actually the same factor. Now that was good because that was a significant step forward. And the next thing she did then was to collaborate with a fellow in Japan, Tasuku Honjo, who was a gene cloner jockey, kind of a guy. And they through the assay, they could identify the gene products that were causing the activity, and from there work their way back to clone the gene. And so that was the genesis, essentially, of what became known then as interleukin 4. So we had interleukin 1 coming from the macrophages. We had interleukin 2 coming from the T cells. Interleukin 3 was not involved in immunology, was more in the hematopoietic end of things. They didn't know it when they found it. But now we had interleukin 4. And interleukin 4 had both B cell growth factor activity as well as B cell differentiation factor activity. And so that was sort of, it was a long story, took a long time, and finally we got some light at the end of the tunnel. And then Eva Severinsen collaborated with another Japanese guy by the name of Kiyoshi Takatsu, who was working with another factor that seemed to work on B cells. At least it worked on mouse B cells. And they got together then with Honjo, Tasuku Honjo, and they cloned a gene for another activity that ultimately became called interleukin 5. And interleukin 5 would also work on mouse B cells, but for whatever reason did not work on human B cells. It caused the human cells, bone marrow samples to be a growth factor for one of the other white blood cells. And then finally, another Japanese investigator, two of them actually, Kishimoto and Toshio Hirano from Osaka, were working independently. Everybody was working independently and competitively in all this business. And even though they were, from the same town, they didn't really share all their data. And so, anyways, Hirano and Kishimoto identified another factor that seemed to help the antibody forming cell whole thing happen, and that they cloned the gene, they purified that, they cloned the gene for it, and that became interleukin-6. So that was all that happened. 4, 5, and 6 happened in 1986. And actually, they had a meeting over there in Osaka in 1986, which they invited me to. I remember that it was 86 because we were at the meeting, and it was in Osaka when the Space Shuttle Challenger blew up. So there's only one other part to the story, and that is going back to Avrion Mitchison in 1971, who came up with the idea that the cells had to contact one another. The T cell had to, in order to help the B cell, it had to actually touch the B cell. And what happened was a whole different other group of investigators identified a molecule on the surface of B cells that ultimately, and they developed, or they identified this molecule because they were able to generate a monoclonal antibody that reacted to the surface of B cells. And they identified this molecule, they called it CD40. There was an investigator by the name of Jacques Blanchereau who was trying to grow B cells long-term, like we had grown T cells long-term. And it turns out, and everybody tried this with all the conditioned media and all this other business, but it never really worked. But if he used the antibody to the CD40 molecule on the surface of B cells, and he used interleukin-4 together, he could get B cells to grow long-term. And that was important. I mean, that was, to me, I was going to say, as important as getting T cells to grow long-term, but I don't think I can say that. It's almost as important. And that was in 1991, and then in 1992, so they knew then that there was this thing on the surface of B cells that was giving a really good signal to the B cells, so that they would proliferate. But nobody knew what was going on there, and actually 4 different groups simultaneously in 1992 all came up with an antibody that would react with helper T cells that would block the ability that would block the ability of T cells to help the B cells through their linked recognition or their cognate recognition. And so that really closes the loop, brings the story back together again. And the molecule that they, on the surface of the T cell, that they called CD40 ligand. And the next thing that happened that really sort of nailed the coffin down right was that in 1993, there were individuals that had a weird congenital syndrome that was called X-linked hyper-IgM syndrome. And the patients could make IgM, but they couldn't then go on and switch the isotype of the antibody molecule from I to IgG or A or E. So they seem to be blocked in a certain stage of differentiation. And essentially that they had mutations in their T cell compartment in the CD40 ligand molecule. And that then solidified the idea that you have to have the linked recognition, but you also have to have these soluble molecules that are coming from the T cells And I haven't spoken with the macrophage contribution yet. We could do that in another chapter. But that rounds out the whole thing with regard to T cell help for antibody formation. And I'll stop there.